prevent for using SPS, as you show here, as the chemical structure. These are the blood bottles for anaerobic, as you show here on the left, and here the white aerobic blood culture bottles. Okay. Applying subculture syringe and drip methods, as you show here, the aerobic bottles, in both of them, gram stain is very important to detect the causative agents, either gram positive or gram negative. The result of a gram stain give me a preliminary test for the culture and the suspected, the suspicion. What's the suspicion of the causative organism? The gram stain. After that, the bacteria from the aerobic and blood from the aerobic bottle will inoculate into blood agar, chocolated Mekong agar, and in anaerobic it is inoculated into the blood agar. Okay. These are the incubators for a blood culture. Okay. How to culture an intravenous catheter trip. كيف ندخل intravenous catheter trip؟ طبعا يعني بشكل واضح جدا. شوفنا كيف يصير عندنا when colonization of an indwelling catheters. هذا indwelling catheters المدفوع بالداخل is suspected of being the focus of septicemia. The catheter tip may be cultured to determine its status. احنا عادة مجرد هو احنا الكاثيتر مجرد ندخله واحنا بدنا نخلق الانفكشن، لكن طبعا هو اكيد من الكاثيتر هو الاستخدام الطبي المهم له، لكن بنفس الوقت احنا هو نصنع we are create the infection itself. اللي هنا شيء يصير حتى اذا يعني بديت اقلق انه ممكن يصير عندي بكتيريا او سبتيسيميا due to catheterized infection، نعمل من هنا شوف هذا الكاثيتر شو يصير. الكاثيتر tip may be cultured where into the culture media. After overnight incubation, the colonies are counted. Above the culture result with greater than 5 CFU, 5 colony forming unit per 1. هاي الطريقة نقطع الكاثيتر ونزرعه بشكل 5 سنتيمتر. اوكي، بعدين نعمل طريقة rolling 4 to 5 times across the surface of the agar plate. Okay, when we showed more than 5 colonies, it is considered positive. The interfering factors patients on antibiotic therapy is the most interfering factors for the successful blood culture. Turn around resulting reporting any isolated organism will be reported. Why? Because blood is sterile body fluid, and any organism must be found. It's considered pathogens. But we must differentiate between contaminant and true pathogen. When we have different colonies with similar morphology, it is indicated it's true pathogen. But different colonies or many colonies with different morphology, that is considered contamination. Turnaround time. Initial blood culture result will be reported as soon as it shows growth. Final results with sensitivity will be issued after 24 to 30 hours of final initial report. Negative results will be issued after 10 days of culture submission. The examination of blood culture. First condition for incubations. After incubation, the bottles after incubation, the plate at 37 centigrades with cap of the culture media losing 5 to 10% carbon dioxide, because certain blood pathogens are carbon dioxide dependent, like H. influenzae, Streptococcus pneumoniae, and serum meningitis. The blood culture incubated for 5 to 7 days unless it's specifically indicated by the physician, like Brucella. Brucella يحتاج maybe reached 1 month. Okay. In manual system, the blood culture bottles are examined 2 to 3 times after for the first, second day of daily, thereafter for a week. The number of blood specimens that should be drawn for culture and the blood time over which they're done depends in part upon the severity of the clinical illness. In hyperacute infection like gram-negative sepsis with shock of or staphylococcal sepsis, It's appropriate to culture 2 blood specimens obtained from different anatomic sites over a period of 10 minutes. In other bacteremic infections, like subacute bacterial endocarditis, 3 blood specimens should be obtained over 24 hours. Okay. Appearance of a broth. How can I suspect the type of microorganism according to the appearance of a broth in the blood culture? If we show uniformly homogeneous turbid layer above the red blood cells layer, gas bubble may be found. It is indicate the gram negative roots. من نشوف البلاد كلتشر بروسس طبعا راح يصير الار بي سي sediments راح تكون in the bottom of the bottles. لذلك من نشوف uniformly homogeneous turbid layer طبعا uniformly homogeneous turbid above the red cell layer, it's considered gram negative roots. In case of meningococcus and pneumococcus, meningococcus mean Neisseria meningitidis and pneumococcus pneumonia caused by Streptococcus pneumonia. Meningitis caused by Neisseria meningitidis and pneumonia caused by Streptococcus pneumonia. It produces less turbidity and usually accompanied by greenish tint in the media. نشوف بشكل واضح green discoloration أو greenish tint موجودة بشكل واضح بالميديا. هذا معناها احنا عندنا ألفا هيموليتيك ستريبتوكوكاي من النيموكوكاي ممكن نشوفها بشكل واضح أيضا. In case of streptococcal infections, we can show cotton ball colonies. كرات قطنية colony تشبه الكرات القطنية موجودة على on the top of the red blood cells. بنشوف هيك شكل cotton ball colonies, that means streptococcal infection. While the upper layer of the media remain clear. This occurs in case of subacute bacterial endocarditis due to alpha hemolytic streptococci. If the causative organism is beta hemolytic streptococci or any other alpha hemolytic organism, hemolysis of the blood will appear together with the turbidity. And in case of staphylococcus infection and due to Staph aureus produce coagulase enzyme, thus jelly-like coagulum throughout the broth due to coagulase production from Staphylococcus aureus infection. Thus, jelly-like coagulum appear in the broth of blood culture bottles suffering from staphylococcal infection. The interpretation of positive blood culture. Virtually any organisms, including normal flora, can cause bacteremia. A negative culture result does not necessarily rule out the bacteremia. طبعا لأن ممكن يكون false negative results occur when pathogen fails to grow. من يكون number of microorganisms is low. من يكون patient under antibiotic therapy. كلها تعطيني false negative culture. A positive culture result does not necessarily also indicate bacteremia. ممكن يكون contamination. واللي هو transient bacteremia ممكن contamination جاي من السكين during the blood specimen collection, or the major source for contamination in the blood culture. Gram-negative bacilli, anaerobes, and fungi should be considered pathogen until proven otherwise. The most difficult interpretation problem is to determine whether an organism that is usually considered normal skin flora is true pathogen. Now, what are the limitations? شنو المحددات للبلاد culture? 3 negative sets of blood culture in the absence of antimicrobial therapy are usually sufficient حتى نقول هذا to exclude the presence of bacteria. إذن حتى نستثني وجود البكتيريا نقول انه ما عنده هذه البكتيريا، يعني يكون عندي 3 negative blood culture bottles والبيشنت ما ياخذ انتبيوتيك، يعني under standard condition فهذه exclusion للبكتيريا. One set is seldom over sufficient. Pro-antibiotic therapy may cause negative blood culture or delayed cone. Blood culture from patients suspected of having Brucella or Leptospira must be requested as special culture. طبعا ليش؟ لأنه احنا عندنا البروسيلوسيس والليبتوسبيرا انفكشن تحتاج specific or special culture condition like carbon dioxide, enrichment media, selective media, and extra. Yeast often are isolated from routine blood culture. However, if yeast or other fungi are specifically suspected, a separate fungal blood culture should be drawn along with each of the routine blood culture specimens. All of these are limitations for the blood culture. Okay. Mycobacterium avium complex is frequently recovered from the blood of immunocompromised patients. In immunocompromised patients, we can easily show the Mycobacterium avium, particularly those with acquired immunogenicity syndrome. The AIDS special procedures are required for the recovery of these organisms. This represents the Castanida biphasic medium. Where I can use Castanida? I can use Castanida for the culturing the blood from the patients with high suspicion of brucellosis, because here I have two types of media: solid media, as I show here, and liquid media. For example, brain-heart effusion agar, brain-heart effusion broth. Thus, when we inoculated the blood from here for 5 ml, occultation of the bottles and incubated at the incubator, after 15 days, no positive culture will remain after 15 days. The advantage for use Castanida blood culture system here for brucellosis, firstly, to reduce the risk for transferring the brucella to the brucellologist or laboratory man during opening the cup. Second, and to reduction the incubation time from 1 month, well standardized بالبروث only إلى 15 days, يعني half the time. Okay, this Castanida biphasic medium. Why it's called biphasic? Because two phases of culture media, solid and broth, is observed in the Castanida. That is all regarding the blood culture. Dear students, I hope you are interested in this lecture. Please do not hesitate if you for any inquiries. Thank you again